materials moving into and out of the cell or around the cell, also towards the end here. There are a number of different processes that make the cell selectively permeable, and some of them are due to the semi-permeability property of the membrane. So the first is a passive process. Passive processes do not require ATP. They are things that happen because the membrane is, by its nature, semi-permeable. They would include osmosis, facilitated diffusion, both of which are really forms of diffusion. So if you see diffusion, osmosis, facilitated diffusion, you're moving across the cell membrane because of a concentration gradient and the ability to get across the membrane. Sometimes you need help, and if you're talking about water, it's osmosis. More on that in a few minutes. I'm going to talk a lot about filtration. Filtration is an important process, but it's really not a movement into and out of a cell. Filtration is more the movement of materials across structures, and we will see it in the kidneys in particular. There are also active processes or physiological processes. Those processes require the investment of energy, and that energy, as always, is in the form of ATP. And that would include active transport, endocytosis, exocytosis. Particularly in the digestive system, we'll see cells that do endocytosis on one side and exocytosis on the other side. And when you move material in and across and out of the cell, it is called transcytosis. So diffusion works for small things, atoms, some molecules, probably about 3 atoms size molecules. Ions can move by diffusion, and diffusion itself is the movement of things from high concentrations to low concentrations. It occurs due to the inherent movement of molecules themselves. Remember, electrons are always in motion around the outside of an atom, so that atom itself is in motion. The molecules that make it up have that Brownian motion, we often call it. But in order for diffusion to work for a cell, to use it as a passive form of transport, the material that you're diffusing has to be able to cross the membrane on its own. Oxygen can do that. Carbon dioxide can do that. There are some lipids that can do that. This makes it a passive process. There's no ATP required. So if you have a membrane that is permeable to a substance, and there is a gradient of that substance, that material will eventually, by diffusion, move across the membrane, and it will continue until you have an equal amount of that material on both sides of the membrane. We call that dynamic equilibrium. Material is still moving in and out, but on average, for everyone that leaves, another one will come in. Facilitated diffusion is very similar, requires a concentration gradient, but the material that you're moving won't cross the cell membrane on its own. So there has to be a channel, a pathway, some sort of transmembrane protein that will help that material move across. And we use this for substances like sodium ions, potassium ions, chlorine ions, glucose, individual amino acids. It's still a passive process because it doesn't require any energy to change the shape of the transport protein. And the energy itself is coming from the concentration gradient. So it's still a passive process. It's still diffusion. The only difference is you need a protein to help the diffusion occur. You need to facilitate the diffusion of that material because it won't cross the membrane on its own. If we're talking about water, then the type of diffusion we use is osmosis. It's not quite that simple. No ATP required. Water will diffuse across the membrane at the molecular level. There are also channels that can help water cross the membrane, so it's both a diffusion and facilitated diffusion type, but the bottom line is that water will move down its concentration gradient into or out of cells, and that will be important when we talk about water balance. So it's a passive process, no ATP is required, and it will go from where there's a high concentration of water to a low concentration of water. That leads us to discussions about what kind of environments a cell could find themselves in, because that osmosis will create what we'll refer to as osmotic pressure. The water on its own will move from high concentrations to low concentrations. So a cell could find itself in a liquid that is isotonic. It has the same osmolarity as the cell itself. There's no water gradient. Concentration of water inside and outside is the same. Water will continue to move back and forth. It's still dynamic equilibrium, but there's no net change. A cell might also find itself in a hypertonic solution. Hypertonic solution has a higher osmotic pressure than and water is at a higher concentration inside the cell than outside the cell. So the water will leave the cell and the cell will shrivel up, as shown here in B, a red blood cell. Hypotonic solution is the opposite. In a hypotonic solution, we have a lower osmotic pressure in the cell, so there's less water inside the cell than there is outside the cell as far as the percentage goes. So that cell will swell and eventually it will burst. This cell in C here is not burst, but it will if it left alone. This can get a little confusing for new students because hyper generally means higher and hypo means lower. And it still does in this case, but tonic doesn't refer to water. Tonic refers to what's dissolved in the water. So in a hypertonic solution, there's a lot of dissolved material, which means less percentage of water. In a hypotonic solution, there's less dissolved material, so there's a higher percentage of water. Filtration is a movement by a force, a pressure. It's used to separate solids and liquids from solids from water, or small particles from large ones. Blood plasma leaves the capillary, water, small solutes are filtered, large plasma proteins remain there. It is a passive process, no ATP required. We won't dwell a lot on it here. For active transport, you're going to have to pump materials from one side of the cell membrane to another. We're going to go from, when we do that, we can go from regions of low concentration to high concentrations. We can go against the concentration gradient, but it's an active process. It's going to require the use of ATP. It's going to require cell energy. Sugars can move this way. Some amino acids can move this way. We also have examples of calcium, hydrogen, sodium, potassiums. In fact, the sodium-potassium pump is going to be the most studied, longest known, and most used example of active transport. In endocytosis, we're moving a large amount of material through a vesicle into the cell, and there are three different major forms of it: pinocytosis, phagocytosis, and receptor-mediated endocytosis. In pinocytosis, the cell will grab little pockets of the liquid that surround it, little water solutions around it. It's often thought of as cell drinking. In phagocytosis, you're going to engulf a large particle. Could be a bacteria by a white blood cell. And in receptor-mediated endocytosis, there are specific receptors for substances that we're going to try to bring into the cell. You're going to see a lot of receptor-mediated endocytosis when we talk about the digestive system. Your individual nutrients come in, a lot of them by receptor-mediated endocytosis. The materials come in, and in the case of phagocytosis, they will be merged with a lysosome. The phagocytized particle, the vesicle called a phagosome, will merge with the lysosome. The lysosome will release its digestive enzymes, break down the material inside that phagosome, and then a lot of that will be released back out into the environment. But not all of it. keep that in mind when we get to the chapter on immunology. Exocytosis is really endocytosis in reverse. We're going to release substances from the cell. So the vesicles that contain the particles will fuse with the cell membrane and release the cell contents, or the contents of the vesicles. And we're going to see this in neurotransmitter release from nerve cells. Nerves talking to muscles, nerves talking to other nerves are going to involve the exocytosis of the neurotransmitter. Transcytosis is a combination of endocytosis and exocytosis. And again, we'll see a lot of this in the digestive system. The materials will bind to one side of the cell, the luminal side of the cell, the side that's pointed towards the digestive tract. The materials will be picked up, endocytosed, moved across the cell, and be released from the other side of the cell into the blood, where they will be taken to the liver and used. HIV also uses this mechanism to get across the membranes of cells in the anus or vagina.